Today we're going to go through topic 6.8, biotechnology. Our learning objectives are going to be to explain the use of genetic engineering techniques in analyzing or manipulating DNA. So in this class we do 2 biotechnology labs. We do bacterial transformation, which is an example of manipulating DNA, and then electrophoresis, which is an example of analyzing DNA. Important vocabulary to know. We have genetic engineering, which is the process of manipulating genes and genomes. There's lots of different ways to do genetic engineering. Our bacterial transformation lab would be an example of that. We have PCR, that stands for polymerase chain reaction. This is a method used to amplify a particular piece of DNA without using cells. So if you've got a small sample of DNA, this is a way to make an artificial way to make more of that exact DNA. And we have recombinant DNA. This is DNA artificially made from different sources, often different species. So in the bacterial transformation lab, we use the pGlow plasmid. That pGlow plasmid is an example of recombinant DNA that has been artificially made in the laboratory, and we've got genes from different organisms represented. Then we have restriction enzymes. These are used to cut strands of DNA at specific location, and we get these from bacteria. Bacteria use them actually to help them incorporate plasmids. But in biotechnology, we use them specifically with our electrophoresis to cut pieces of DNA at specific locations to give us different fragments. And then plasmid. This is a small circular piece of DNA, very commonly found in bacteria, occasionally found in eukaryotes, that's separate from the main chromosome. So when it comes to sequencing DNA, which is our method of analyzing, we've got 2 very common ways. So there's electrophoresis and then whole genome sequencing. So here we're looking at electrophoresis. Again, this is a lab we do in class as well. This is going to separate macromolecules, so things like DNA and proteins, based on their size and their charge with an electric current. So here we have a basic sort of diagram of how electrophoresis works. So we have our solution here, which helps with our electrical conductivity, our gel, and then little pockets or slots in our gel containing our DNA samples. So the samples get put in the wells. Then we turn on the electricity, and we're going to have a negative electrode and a positive electrode. Now, DNA is slightly negatively charged. So once the electricity is put on, the DNA is going to move away from the negative electrode toward the positive, opposites attract. And as it moves, it's going to be separated out by size. Smaller molecules are going to move further, and they're faster than larger fragments. So when we have this run in class, we'll have the electricity going for 10, maybe 15 minutes, and we'll see our fragments of DNA, and those fragments are made with restriction enzymes, separate out by size. We have our longer bands are closer to the negative, the starting line, and the shorter bands closer to the positive. And when we do this, oftentimes there will be a ladder, or sometimes called a marker, where we know where our fragments have known sizes, and so we'll be able to compare knowns with our unknown fragments. In class, or in our laboratory, we're going to use an electrophoresis chamber that looks exactly like this one here. And so we can see here's our negative electrode, positive. Here's all the wells, and this is a micropipette that will be used to add these samples to it. Then we have genome sequencing. So this is figuring out the order of the DNA nucleotides in a genome. So this is knowing the exact type, all the letters in your bases. So this process is very expensive, takes a long time. and something where there's lots of advancements in. Particularly, we need advancements in computer science, just that computational ability. Just recently, I read an article about a worm genome that was sequenced, and it took over 100,000 hours to sequence it. So this is a lengthy and expensive process. Now, particularly medical science is hopeful as this process improves. It gets cheaper and quicker. That'll help lead eventually to customized medicine. So some applications for DNA technology. So diagnosis of disease. So particularly with virus genomes, because virus genomes are very small, we can do whole genome sequencing to see all those differences to know exactly what virus somebody is sick with. This has been in the news a lot recently with COVID-19. We now know that the COVID virus is continuing to change and mutate. And so there are 2 new variants that have been sequenced, one from the UK and one from South Africa. And scientists are studying those, and they know where that difference is because of whole genome sequencing. They know all the letters that are made up in that virus's genome. Also used in forensic applications, which is what a lot of people tend to be very familiar with due to crime shows, things like CSI. So that's when we take a DNA sample and we can match it up, sample from a suspect, match it up to DNA collected from a crime scene. In our forensic applications, PCR is going to be very important because oftentimes the amount of DNA collected is very small. So we need to amplify it to create more DNA in order to analyze it properly. Next, environmental cleanup. So not just in medicine, not just for crimes, but the environment can also benefit from DNA technology. There's research being done into using microorganisms to help clean up, removing metals from mining sites, cleaning up sewage, cleaning up pollution, particularly oil spills. There are some microbes out there that naturally will break down and consume oil. And so there's technology in trying to improve upon those bacteria so that they are useful for bioremediation, so a more ecologically friendly way to clean up our pollution. We also have environmental DNA, or sometimes called eDNA, and that's genetic material that's obtained directly from environmental samples without having to have the organism around. All living things, we're constantly shedding cells, losing DNA to our environment. And so environmental DNA is really important to ecologists trying to study biodiversity. In the ocean, our marine biologists get a lot of information from this. Oceans are too big, especially when you're trying to study biodiversity and how many different species are out there. But through using eDNA technology, they can take a sample of ocean water, separate all the DNA from that, analyze all of that DNA, and then that gives them an idea of how many species were in that area based on their leftover traces of DNA that were present. So when it comes to genetic engineering, lots of uses in medicine, particularly with gene therapy. So that's changing the genes of someone who has a genetic disease. So we've talked about some genetic diseases in this class, like cystic fibrosis and sickle cell anemia, which are due to cystic fibrosis. It's one, or both of them, it's one amino acid that's wrong. It's just one gene. So the hope is with gene therapy that we're able to change that person's gene in order to cure them of their disease, instead of our current method of just trying to moderate and reduce their symptoms, or having them go through something super extreme like a bone marrow transplant. And there are some articles linked here for more information on gene therapy. And then we've got Also useful for the production of pharmaceuticals, so medications, being able to use gene splicing and cloning in order to get bacteria to make medications for us. Things like insulin, we currently use bacteria to produce human insulin. So we've got genetically modified organisms, or GMOs, very common in agriculture. Although the E. coli that we're going to produce in the bacterial transformation lab are technically a genetically modified organism. So in agriculture, it's used a lot of times with, or particularly with plants, to insert desirable genes that help farmers to increase their productivity and their efficiency. And a couple of common genes that get inserted. There's a gene that makes plants resistant to the herbicide Roundup, so farmers can just mass spray Roundup on their fields. The corn grows, the weeds don't. There's also some that that have the plant making a pesticide, so that way farmers can spray less pesticides on their fields. Our last example, and the most recent one, and one that's used in a lot of, or is becoming very popular now due to ease of use, is CRISPR for doing the genetic engineering, for cutting out that gene and adding in the new one. This comes from a set of bacteria that are used to add and remove information. So now that kind of goes with our gene therapy. So there's an article here about a person getting gene therapy, but they're using CRISPR to change their genes. And so that's something that very recently has come on the scene. It's also caused a lot of ethical controversies. We've got linked here for you. Here's a video about how it works that you can watch. Also linked are videos. This is a discussion on how CRISPR works. And this is a look here, this article, to some of the controversy. There was a scientist back in 2018 that engineered 2 babies to be resistant to the HIV virus. And the way he used CRISPR, it actually potentially is altering their germline, meaning altering their gametes, so they could pass on that change to their offspring.